Study about the structure and function of the DNA. DNA stands for deoxyribonucleic acid, and DNA is a molecule which is located inside the nucleus of the cell, and its main job is to store and code the genetic information of the body. Now, for most of the life cycle of the cell, the DNA is located inside the nucleus of the cell like a coiled noodle. But during the time when the cell replicates, the DNA is arranged in the form of structures which are known as chromosomes. And these chromosomes help to keep the DNA stable during the time of the cell replication. Now, most of you are familiar with the double helix model of the DNA. The double helix model demonstrates that DNA is basically a polymer. Now, to understand the structure of DNA in a better way, you have to understand the definition of a polymer. So the polymer is basically a molecule which is consisting of many repeating units which are known as monomers. If we take the example of a simple polymer called as starch, in this case the repeated monomeric units are known as glucose. These glucose molecules are joined together by glycosidic bonds and they form the polymer which is known as starch. In a similar way, DNA is also a polymer, and in this case, the monomeric units which form the polymer DNA are known as nucleotides. Now, this is the basic structure of nucleic acids, so if you want to learn in more detail, you can refer to that video. The link to that video will be in the description below. Now, the carbons present inside the deoxyribose sugar of the DNA are numbered from 1 to 5 in the clockwise direction. And you should remember there is numbering of the carbon atoms because it will help you to understand the concept of directionality of DNA. Now, what happens is that the third carbon in every deoxyribose sugar forms a bond with the phosphate group of another nucleotide that is present below it. And this bonding extends in both directions. Now, if you zoom into this structure, you can basically see that every phosphate is connected to two sugars. One bond with the third carbon of the sugar above and one bond with the fifth carbon of the sugar below. And now again, if we zoom out, you can clearly see that due to the bond formation between the sugar and phosphates, we create this sort of sugar phosphate backbone on one side. And we have all the nitrogenous bases which project out of this sugar phosphate backbone on one side. So what this basically represents is essentially a single strand of DNA. Now, if you go back to the step ladder model of the DNA, you can clearly understand how the different nucleotides in a single strand of DNA are joined together to form the sugar phosphate backbone, which was represented by these blue vertical lines. And in the center, we had these orange bars which connected these two lines, which are essentially the base pairs from one side. Now, in the opposite side, we have the other strand of the DNA, which is quite similar to this original strand, but has some key important differences for you to understand. And to understand these key differences, you have to understand the concept of directionality and complementary base pairing. Now, what directionality essentially means is that it is the end-to-end chemical orientation of a single strand of nucleic acid. In this example, you are saying the single strand of a nucleic acid. Now, the direction of the DNA is expressed in two ways, 5' end to 3' end or 3' end to 5' end. Now, if you look closely at this structure, you can basically see that it has a 5' end to which a phosphate group is attached, and it also has a 3' end below to which a hydroxyl group is attached. So if we come back to our DNA ladder model, we can see that the strand we made before runs in 5' to 3' direction. Now, in a similar way, if you look at the opposite strand, you can basically note that there is one important key difference, that it runs in completely opposite direction. It has its 3' end in this direction and its 5' end in this direction. So it is said to be running from 3' to 5' direction, which makes the two strands of the DNA anti-parallel. Now, the next important thing for you to understand is the complementary base pairing, which describes the manner in which the different nitrogenous bases of DNA strands align with each other. So it basically means that the base adenine pairs always with the base thymine, and the base guanine always forms hydrogen bonds with the base cytosine. You can see this rule in action in the DNA stepladder model, where you can see that the base A always pairs with the base T and the base C always pairs with the base G. These bases are essentially forming the hydrogen bonds in the center, which holds these two strands of the DNA together. Now the complementary base pairing is very important in one of the most important aspects, that it maintains a proper distance between the two strands of the DNA, which is very important for the stability of the two strands of the DNA. So now if you look at the double helical model of the DNA, you can understand what these two helical lines mean and what the central spokes which are connecting these two lines are essentially made up of. So this was all about the basic structure and function of our DNA.